So from like many days we were talking about Blue Native page already. So now you people know what is Blue Native page. Even that day thylakoid isolation, I explained you why we do Blue Native page and all. And sir also explained you what is Blue Native page. So can someone tell for what we are doing Blue Native page, why we are not doing SDS page? You need intact protein complex, which is photosystem 1 complex and photosystem 2 complex. We need the complexes for studies, so we do Blue Native page. And we want to see in super complex level there is any dissociation, so that the bands will be lesser in intensity or more, whatever the band intensity, at least you can tell PS1 is more, PS2 is more, what is happening with the photosystems. So this is the basic thing why we are doing Blue Native page. And here we won't use any denaturing agent, no SDS, no beta mercaptoethanol, no heating, nothing. Everything will be in ice cold temperature, and also you are going to work with complex, so it should be in dark. Whatever I told thylakoid membrane isolation, Blue Native page also it will be very good if you do in a cold room in a dark condition. Then okay, in SDS page what is the buffer we use to keep the pH? Tris HCl. Here we will use little bit more more refined buffer system, which is Bistris and Trisin. And pH 7 should be maintained in both. 7, right? Not 7. pH 7 should be maintained in both. In SDS page you will have different pH, right? But here you will have same pH, and pH is very important because to maintain the voltage, to run the complexes, this pH will play a key role. So pH of whatever the two buffers, you start BN page first by preparing these buffers. So when you are planning to do a BN page, first you are going to prepare two buffers, which is like Bistris and Trisin. Okay, and the Bistris will be part of all other ingredients or the composition of the gel preparation. Okay, and here you are going to have a gradient of gel, not like in SDS page you will have a single percentage, right? 13% gel, 15% gel, whatever. I think here if you see in SDS page there will be different percentage of page, and it will be fixed in whole system. But in case of Blue Native page you will have a gradient of gel, which is like according to your complex size you can change. Sometime it will be like 4 to 12%, sometime you will vary a bit 3.5 to 12%. These all the percentage will be depend on which complex you want to separate, and depending on the organisms also this will change. Which one? The gradient preparation I will show you. We have a gradient mixer here, and you can't prepare it like in SDS page. You need to make a gradient. So if you just add something with a pipette, you are going to you are not getting a gradient there. Everything will be mixed. So here what we will do, we will have a higher percentage, which if in our case we are going to make a 3.5 to 12% gel. So I'll have the minimum one 3.5%, and we'll have the heavy one as 12%, which is called light and heavy. Okay, this what are the extreme you are going to do. So this percentage, and what makes the percentage of a gel? What you are telling percentage? Like if it is a 15% SDS gel, what is that?
SDS in percentage, 1% of SDS dissolved in
No.
smallest.
No. Okay, pore, which is related to what is making the pore? Acrylamide. So acrylamide is making this pore size, right? So this percentage is basically making the pore size. So which is related to acrylamide. So you change the acrylamide concentration accordingly to make this 13% or whatever I am going to make, you are going to change this only. Your heavy and light you can vary. So first you will try a normal one. So you understand, okay, in this percentage mine is not coming, I need bit more pore size. You decrease the or increase the acrylamide percentage. This is how you make. Okay. And for next what? Okay, we prepared the buffer. Now what all we need in a BN page system? Can you get my copy? Here or here? Here, I think, right? So we talk about the pH now. We talk about the gradient. And if you want to read from the basics to more, refer to Eva Marie Arrow paper. Arrow. EVAMARIARO. And they will have a video tutorial in that paper. If you go to their website, they will have a tutorial how they make the gradient and everything. Because if I start BN preparation today, I'll make buffers. Tomorrow I will make the gradients, and next morning only I will do. So we can't do this possible like a 3-day experiment. So I'll try to give a maximum idea about what we are doing, everything. Now you can go and see this video and read the article. You will get more idea about how to separate and all. Let's go to that mixer and I will just try to explain how we are doing that. Take that also. That. Again we can't. We'll just go and come. It's fine. Maybe 5 people come with me. I'll show what is. Nothing is there. I will just show you this. Maybe first 5 people come with me, or half of you people can come. Yeah. So, see, I wrote here heavy and light. So, what you're going to do, you will connect this pump. This is basically a pump system, so that it will go, you can control the flow here. So, you don't want to just mix everything. You want to go it very slowly so that it will form a slow gradient. I already made this from morning. Otherwise, this gradient making will take again 3-4 hours. So, I came in the morning and I put this because it's not feasible to do this. So, I just connect this to this using a small tip or something. I'll keep it here and I put the different percentage of solution, light and heavy, separately. You should not mix it. It should mix slowly from here. From here to here there is a connection. So, according to that, here you can maintain the flow, speed, everything. Maximum slowly you can do, like here I keep almost 2.3 ml per minute. 1 minute only 2.3 ml will go inside. So this is the flow rate. If you decrease it to 1.5, 2, it will be more good gradient. Slowly something is going, then it will form a very nice gradient, right? So that is what why we are using a mixer. You are not going to put using a pipette. So that work will be done by this mixer machine. So it goes like this and slowly this will control the flow rate. Sucking and flowing rate will be controlled by this mixer machine. That's it. And once you fill up to here, you are going to add normal like isobutanol or whatever, and then after 3 or 4 hours you can see it will be solidified and a nice gradient will be formed. And here APS and TEMET percentage will be very less, not like SDS page in half an hour it will solidify. No, the solidification or the polymerization should be very slow so that the gradient will be formed very nicely. So this is the concept behind this. So we'll keep it, sometime we just keep overnight like that so that it will form slowly and next day only we use this. So once it is done, like a normal SDS page, you can prepare the stacking and load. There APS and TEMET we will be adding more so that in half an hour stacking will be done. So now I just poured the stacking. It will be ready by the time we prepare our sample. Any doubt related to this?
Stacking and dissolving should be separated.
Yes, because you need the comb, right?
Yes.
So pH and all will be same. Every composition will be same. No difference.
With this machine, only the resolving is
Only resolving, because resolving only you will have a gradient. Yes, you can do. It's clear about why we do this mixer and what we are clear?
Below the
Yes. Maybe you will keep always a like space between the resolving and this, right? So at least a 0.5 centimeter here gap will be there. Okay. So maybe next people can come.
Yep. Instead of mixing, you mix both the heavy and light, and then it is.
No. I will open this pump. Then it goes like this. Then I will open the light because you need down the heavy 12%. So you open it, 12% goes slowly. Then you slowly open this. Once it is reaching here, so here they will start mixing. So now whatever it goes will be slowly mixed thing. But once it is going by drop, it will go.
First
How it will be? Something heavy and light. It will go drop by drop, and you are keeping a 4 hours. Obviously, the heavy things will go down, and the will come up, right? So that's what here happened. And also to make this heavy, we will add glycerol in heavy. It will be viscous. So with that glycerol heavy, it will automatically go down. And here we won't add glycerol in light. So that's how this system works.
Reference what I've given, they have the protocol also.
Yes, they have the protocol and they will show how they are doing. There is a video. If you click, you can see that as well. Okay, can you ask the other people to come? Remaining people can come now. So as I told, we are not going to do something with a pipette like SDS page. We need a very nice gradient from 12 to 3%. So I told you a higher percentage and a lower percentage you will be making, like 12 and 3%. And in this 12 you will add more glycerol also, so that it will be heavy. So what happens? This mixer can control the pump pressure. There will be like the sucking and flow rate will be controlled by here. So I keep like a very slow flow rate, that so that it will come like drop by drop, and something is heavier and light is mixed, but you allow them to keep for some time. Automatically the heavy things will go down and the light things will come up, right? So once you add this, you are going to add very less APS and timid compared to SDS page here. So they won't polymerize so fast, so that they will get time to make the gradient itself. And just to make, you can't do like drop by drop here using a pipette. So you use a mixer which can control the flow rate. Right now we are keeping like 2.3 ml per minute. In a minute only 2.3 ml will be going down. So if you see this, it will be like one drop by drop it will go. If you decrease the flow rate, it will be more good gradient. Like if you can make 2 ml per minute, it will be more fine gradient you will be getting. Then yeah, that's it. That's why we use this mixer. 3 to 4 hours maybe it will take time to form a nice gradient. Sometimes we just keep overnight like that, so that morning we can do the stacking. And there is no change in pH or anything. So you're going to use the same composition for the upper part also. You will just do. So today morning I came and I did the resolving part, the down part. And right now we just poured the stacking. But in stacking part we add more timid and APS so that it will solidify very fast. In half an hour you can use it for loading the sample. So all clear with the system. Any doubt? Clear? Okay, then let's go and learn about what is solubilization and how to do that. I think everything is ready. என்ன ബുക്ക് ഞാൻ പിന്നെ തിരിച്ചവിടെ വെച്ചോ. Can you bring that? $700. You read this paper I mentioned, you will get very clear idea. If I start explaining each thing in this, it will take time. They have explained each and every component, why it is important, and even so we isolated the thylakoid already, and the chlorophyll we estimated per microliter it was 0.7 or something, right? So now we need to make, so we are not doing denaturing, and I mentioned already to solubilize the membrane we use some kind of detergents. You remember any detergent? Beta DM, alpha DM, digitonin, these are all mild detergents which is non-ionic, and you can use it for solubilizing the membrane and to get the photosystem supercomplexes. So today we are going to use beta DM, and if you are more specific about working with like PS1 which is very mild and all, you will be, people normally use like alpha DM or more mild one which is like digitonin, because when it is more harsh detergent there are chances this super complexes may get bit dissociated and all. So you can use more mild detergent. So whenever you read paper, when you are concentrating on PS1, people will use, mostly they will work with alpha DM or digitonin. Okay, even I work with PS1 system more, and in my work I used alpha DM when I am like focusing and I need to take out PS1 particle, I use alpha DM so that it will be more stable. When you use beta, which is little bit more harsh, and you are going to separate all the complexes and study, we use beta DM. So today I am going to do beta DM. And when you are going to work and solubilize this thylakoid membrane, there are two major things for the solubilization, maybe three. One is the chlorophyll content, like not chlorophyll content, what is the concentration you are going to do. If a lot of thylakoid membrane, basically if there is a lot of thylakoid membrane and you are adding a very little percentage of beta DM, that is not sufficient to form. Sir mentioned, no, they will form micellae and this will go inside, and it's a soap action what basically they do. So you need to have a particular concentration of chlorophyll as well as a particular concentration of detergent, then only this work in a proper way. And also the solubilization time, how much you are keeping. You will mix this thylakoid with this whatever detergent and you just leave it, it will be completely destable after some time if you see. So this time of solubilization, concentration of chlorophyll as well as concentration of detergent, these three should be in a very proportionate way, then only you get a better result. Otherwise your results will be varying every time. You do very slowly one time, next time you do like you just added and kept it somewhere, then after 1 hour you are loading. This will all affect your supercomplex separation. You should standardize. When you are starting a BN page, you need to standardize the concentration, you need to standardize the concentration of detergent, also you need to standardize the time of solubilization also. So you have to do a standardization experiment first with different chlorophyll concentration you can try. Next you can try with different concentration of detergent. Also you can try with different detergents, beta, alpha, these all people do. And next, last you can do solubilization time. Like I am going to keep 15 minutes in ice and 10 minutes in centrifugation, because once solubilization is done, you are taking out the protein complex. Then what happens to the membrane and all? When you load along with membrane, they won't go. They won't separate in the gel. So you need to remove that. So you have a centrifugation step also. Once solubilization is done, you are going to do a centrifugation to remove the extra particles, membrane, lipids, everything. You just need the photosystem complexes. Okay. So this combinedly, in my case, it will be like 25 minutes plus loading and all, it will be like 5 to 10 minutes. This is the solubilization time. I am getting good result. But this may not be the case of another organism you are going to work with. So you need to work with this system and you need to understand how to change this and all. Then you get a better separation and yeah. Okay. Then book. രാഗേന്ദ്ര സാറിനെ വിളിക്കുന്നുണ്ട്. സാറ് വിളിക്കുന്നു.
Every day.
So we talk about the buffers. Acrylamide I mentioned, glycerol I mentioned. This is so in gel composition there is only a buffer. I mentioned Bistris, and along with ACA, that aminocapric acid you will be adding, and acrylamide, water, glycerol, APS and timid. This is it. We are not adding SDS or whatever beta mercaptoethanol, nothing. Now I mentioned chlorophyll estimation. So now how we are going to do the solubilization. So in solubilization preparation, you need first thylakoid. Obviously that is your sample. Then plus thylakoid, you will add the buffer Bistris, plus glycerol also will be added in that, which is combinedly called Bistris and glycerol, BTH40G, like 40% of glycerol will be there. This is also just to make it bit heavier so that the sample, when you are loading, it will go properly. In lamellae also, we have glycerol. What is lamellae buffer? Lamellae, sample buffer. In SDS page, you will prepare sample, no? The protein sample, you will add lamellae, right? I am thinking you all worked with SDS page. No. So, okay. So, in SDS page also we add this glycerol just to, when your glycerol is a bit viscous and heavy, right? So just to go the things down properly, there is a small well you will have and you will load something. So to the well it should go properly and settle. So you add glycerol. Here also the same purpose you will add. And next is detergent. Whenever you need a protein denaturation, you add a detergent. That is beta mercaptoethanol, or you can use SDS is obviously in the system, but you are not going to add it for the disulfide cut and all. You are going to add beta mercaptoethanol. Another one is also the DTT. Yes, DTT. Either DTT or you will add beta mercaptoethanol. But they are denaturing agent. They are going to completely denature a protein. But here you will, instead of that, you will add beta DM today. So your thylakoid sample, then the buffer, then you can add beta DM. And here again you will add a little bit protease inhibitors. Okay. Then obviously you make everything equal, right? When you have a control and something treated and all, if there is a stress, total protein will be very less in that. Take Chlamydomonas, I am growing in very high light, and something I am growing in normal condition. This very high light, they don't want to work, they are stressed, their protein, complete protein level will be very less. But in control, everything is nice. Protein is, consider 100 is there, whatever unit, 100 is there. But at the same time, when you take the same amount of cells from a treated sample, it may be 70. But now you want to see Rubisco in both. Can you compare this total protein of 70 and here total protein of 100? Can you compare now? No. Because now the total is very less here. So how you will compare? What you have to do? You have to make both 100 or you have to make both in a 70, right? Or you can make it 50. Then you compare Rubisco level. Then you understand, okay, even though total protein is this, Rubisco is upregulated in this or downregulated in this, because different proteins will have different expression in different stress condition. This is why we always keep a baseline, right? That is why we estimate protein. That is why that day I estimated chlorophyll. I explained you photosystems, why we are doing chlorophyll. You have to make equal photosystem functioning. You don't want to take 2 photosystems from here and control is having 100 photosystem. Okay, let's see the efficiency. Obviously, there are only 2. How they will be efficient? You need to make it equal. By your isolation, you can take more and keep it equal. Then do your experiment. Then only you understand, right? So here also we are going to do equal chlorophyll. Whatever, how many sample you have to make it equal chlorophyll. I have a 500 microgram per microliter, or consider I have like in one sample, in my control I have 2 microgram per microliter, and in my treated I have 1 microgram per microliter. Okay, and I want to make it, how can I make it equal? How much volume I should take? 1 microgram in stress condition, 2 microgram I have in concentration. I am telling. When I am telling concentration, I should not tell 1 microgram. I should tell 1 microgram per microliter, 2 microgram per microliter. So consider I have 1 microgram per microliter concentration here and 2 microgram per microliter concentration in the other. Then how can I take equal chlorophyll? How much volume I will take? Half. Either one I can do, right? If I am taking 10 microliter here, I should take only 5 microliter here. Then I can add little water in this. So the volume will be equal. And then I add beta DM, both in equal. Now photosystems are equal. I am comparing something equal. If I directly take 1 microliter from each, what will happen? Here only half photosystem is there. So this is the concept. Always you should remember. Okay? Okay. Even if you are doing with equal protein also, same. I have 2 units of protein here, 1 unit of protein here. I should make it equal. That's it. Okay. And I just told no, we will add little bit of water. You can use some resuspension buffer also. That day we resuspended in TRB2, right? So the same buffer you can use here to make it equal. The volume will be high. That's it. Now do one calculation. We have 0.7 microgram per microliter, right? Thylakoid. So I want to take 100 microgram chlorophyll. So how much I should take volume? We have a concentration 0.7 microgram per microliter. So I need 100 microgram chlorophyll. Nothing is there. How much I should take? How much microliter? 0.7 into in 1 microliter 0.7 microgram is there.
Approx 142.
How?
Because in 1 microliter 0.7 microgram.
Yes.
There.
Yes.
So, 100 microgram it will be
100 into 100. Or 100 divided by 0.7. How much you got? 142. So I need to take 142 microliter whatever thylakoid we have. If I want to make a preparation of 100 microgram of chlorophyll concentration thylakoid to be solubilized, right? Okay. So 0.7 divided by 100. If I need 100. If I just want to make a 10 microgram preparation, then it will be 0.7 divided by 10. That much volume I have to take from that. We have an alicot of 10 by 0. So we have that day 100 microliters volume here. If I need 142, I need to make like 2. So here the problem is, whatever the thylakoid we isolated, we should always keep a chlorophyll concentration. For me, it should be like 2 to 3 microgram or 2 to 4 microgram at least. But whatever the thylakoid we got that day is very less in concentration, which is 0.7 only. So I will just make a preparation today for demo purpose, but whenever you want to do. So if I want to make it more concentrated, what should I do now? This is 0.7. I want at least 1 microgram or 2 microgram. What I can do? What is the possible way? How? If I increase the volume, okay, I have here. Initially we need to, but now I can't, right? What we can do? What is resuspended? How we resuspended this? Buffer. If I remove little buffer, it will be more concentrated, right? So I can put it in centrifuge, do the same what we did the centrifugation. If you remove little buffer and again resuspend, it will be more concentrated. So per microgram, it will be 1 or 2 microgram you can get. If I take half of this one, it will be 1.4, right? Because now we have 0.7, right? If I take a half buffer and mix it whatever the remaining, the concentration will become double. So I get a 1.4. But now we are not going to do anything. I will show you preparation, maybe using a 50 microgram. I'll take half and I'll show you the preparation. These cases can happen. Sometime when you work with stress condition and all, definitely this will happen. You will get a very less concentration. You just have to do the centrifugation and make it more concentrated. But these are all not recommended, because it's not good for the thylakoid, it's not good for the photosystem. So when you are planning to do, make it very thick slurry, not like too much buffer you should not add. That's it. Okay, now I will show you. First one we need is thylakoid. I have thylakoids. Then we need BTH, I told you, which is basically buffer, bistris and glycerol, just for making it the buffer system, and glycerol. Then we have, this is inhibitors. Protease inhibitors. This I will not explain, because during thylakoid isolation I told you why we are adding. This is, whoever did SDS space, they must know why we added dye. Visualizing. So here also same purpose. Look at there, that is BN page. Can you see the blue color, how it is coming and all? In the upper part it will be more greenish because it is photosystem complexes. When it comes to down, they are not more with chlorophyll. So if you want to visualize them, you need some color. This is Kumasi only. Okay. G250. Yeah. Kumasi mixed with the bistris and whatever the buffer system. And then what we need? Then you need to make everything equal. If I am going to work with 2, you have to make it equal. So either you can use a resuspension buffer like TRB2, or you can use some nice good milky water, good grade water also you can add, no problem. Both will work. So and for me, the solubilization time I told 15 minutes, and it will go, can you keep it 4 degrees? And everything should be ice cold, and no lights will be on when you are doing in a proper way, because photosystem will get exposed, your entire result will change. Sometimes the LHCs will go, that all know what was that? LHCs will go and bind to PS1. What is that? State transition. So he is going to get the quiz. So state transition will happen. So if I am working with some stress condition, like salinity or something, there is no state transition. But I exposed to light. They are functional now. They don't have any problem. They are functional. You can work. Something happened here, state transition. Whatever the BN page you will see is not the result of salinity. It will be like because of the light. So you should do it in green light or dark. Got? So we will prepare 2-3 vial and I will give it to you people. You can do the loading. And next after this, okay, now other things we missed is the running system. So in an SDS page, we use running buffer, right? But here you will have an anode buffer as well as a cathode buffer. It will be separate. Okay. And okay, can I refer this and tell you later? I am also not confirm about this. Yeah. Yeah, anode and cathode buffer. In cathode buffer, you will do 2 times. First, you will use this blue color solution, and later, this is also Kumasi only, same. And in the second half, you need to see it more like if we run, you will see. You will remove this, and this is like more. It won't be exposed. The proteins or whatever you are loading in this color, it won't be exposed. And this Kumasi has the power to bind and carry out the running. So this we use till the half of gel run, and later you change this to a plain one so that you can see the separation and everything. Yes. Staining you can do later. Again, you do staining and destaining. This is not staining and destaining, but you just change the buffer. That's it. So this is the 2 buffer. In SDS page and all, you use in the same pH, same buffer, but here everything is pH 7, but you have an anode and cathode buffer, and basically this is also made by Bistris and Trisin mix. Cathode will be Bistris and Trisin, and this will be Bistris alone. Then wait, I told something wrong. Yeah. Bistris alone only anode, and cathode will be Bistris and Trisin composition. I don't have to tell you in detail how much we are adding and all. This is unnecessary. If you get a protocol, this you all can prepare. That's it. Then what we missed? Running a gel. We prepared the buffer. We prepared the gel. I explained you the solubilization how to do. And then for running you need that. Okay, one more important point. You have to run now the gel. So you need to give a voltage. And here voltage also plays very important role. People do in 2 ways. Some people keep a constant ampere and it will run. Here we change the voltage. Okay. And the ampere should be maintained like 2 milliampere. And the voltage I will increase. We keep constant ampere and change in voltage. Some people do constant voltage also. And how I do, I start with a low voltage of 60 volt. Then I increase after half an hour, I will increase to 70. Then I increase to 80, 90, 100 in half an hour gap. And by 2 hours it will be half completed. Then we change the buffer system inside. The cathode will go inside. We change this, and what we do then, we increase the voltage, and the gap will not be half an hour, we will keep for 1 hour. So like this, almost 4 to 5 hours you have to gradually increase the voltage and reach to 200 or 250 volt, it will be completed. So almost 5 hours it takes for complete running.
After changing the buffer again from low to high voltage?
No. We will now only increase the voltage. We won't start. It won't run in that voltage. 60 volt if you keep, this heavy complexes won't go down. The current should be more. That's it about explaining. Now we can prepare the solubilization. Any doubt in this? Then after that you can take the directly an image, you will get some image like that. I will also show you some images in PPT. What all will happen if you do something wrong and all, I will show you. And then you can stain this with Kumasi and you will see more band. If something free, new proteins, LHC protein, something is there, you will see this Kumasi will go and bind. You can do staining of this also, and normal destaining all you can do. But first of all, you get a picture like this because they all are already pigmented. And whichever is not pigmented, this Kumasi will be pigmenting them. So,
After changing the buffer system to anode, same step should be followed.
no, we will increase 150, 200, 250. Append of tube. Or 3-4 append of tubes.
Experiment will take till 8:39, no? 5 hours.
No, I am thinking 5 hours running we will keep. It will run. And tomorrow I can show you the result. Okay. Till loading and keeping in cold room I will show you. After that you people don't have to stay. We'll see how it is running. And again, as I said about thylakoid isolation, this also you have to do eyes dark. You should be fast, but again you should be very gentle because you are going to work with the native complex and membrane. Should be very fastly doing, but you should not make like foaming and you are going to add a detergent. If you do like too much pipetting and all, full foam will be coming. This is not going to help you. Everything will be unstable. So you people can also add and try. Maybe we'll get one more ice. This is the only thing they are going to do. Let them do. Maybe one more. Shreya. Okay, one more you bring. Let them prepare. So make like 3 groups, and this 3 group people can at least do the solubilization part just for practicing. Or okay, let them bring the ice. I'll tell you what, at least you will have an idea what all you are going to add and all, but I won't tell. This will give you a good result because you will be slow for sure, and that if you don't maintain the time, adding each thing, if you don't maintain the time, everything will be ulta. You won't get the result. If you are late by 2 minutes, 5 minutes also, your results will be little bit change, will be there. Also, if people work with mitochondrial complex, they use the same native page. There are also complexes you are going to work. So whatever protein complex if you are going to work with, you can use native pages.
In today's class, Takashi sir was asking some question related to fluorescence kinetics. In that poster, kinetics.
Which one? 77K CD, which one? Today's whatever he showed is HandyP you people did, no? HandyP, how you got the data? Like he must showed like this, and he must explain how log scale he changed and all. If you don't change, you will get a kinetics like that. If you change that, you will get like this. That's it. Okay. Now, yeah, whatever. Yeah, one by one they can do. They are just doing demo, right? whatever we have, we don't need 1 ml, we need 10, 20. How much we have, just give them, let them do. So today we won't be maintaining the time and all, but at least you will get an idea what all we are adding inside this, what we are doing and all. Okay, so I'll give 2 Eppendorf tubes. Who is going to do? So we have 0.7 and you got like 142, that is for 100 microgram. But when you're loading, you are not loading 100 microgram of thylakoid. You can't, like chlorophyll concentration, you can't, it won't run anything. A normal range is like maybe 0.8 mg or like you can say 10 microgram. In a well, you're going to load only 10 microgram. Okay. So from this 100 microliter preparation, okay, so normally what I do, I make a total of 200 microliter. The preparation in this will be 200 microliter. In this 200 microliter, I will have thylakoid of 100 microgram, whatever the volume, maybe 20 microliter will give 100 microgram. Now 142 you told, right? 142. So normally we will have more concentration, so I will take a very less volume of thylakoid, maybe 20 or 30 microliter will give me enough. It will be very concentrated. And I will add BTH. Here you can add BTH will be 100 microliter. This will make it heavier, right? The glycerol part. And you need inhibitors. You will add 10 microliter of inhibitor, whatever I'll give you the inhibitor. This is already a cocktail. You can directly add 10 microliter. Then beta-DM, I already prepared. This is 10%. So I need like 1%. So in 200 microliter, how much I should add? This is 10%. I need working 2%. And the volume is 200 microliter. So how much volume I should take from this?
How much concentration will be?
We need like 1%. How much it will be? 20 microliter you need to add. From this 10%, you take 20 microliter, add to 200 microliter total preparation, it will become 1%. So I am using 1%, but we'll try from 0.5% of beta-DM to, you did something. Again he is doing something. So this is 10% stock of beta-DM. So yeah. So I'll take 20 microliter. Now 20 microliter beta-DM I added. BTH 100 microliter. Then inhibitor 10 microliter. Then what? Thylakoid. Now we are not using 100. We will use only 50. But that is not the ideal. I prepare always 100 microgram. If you have very little, you can prepare 50 also. No problem. But again, your loading volume will increase. I know this is too much things. But let's get some idea. And later you can work on it. Okay? But I'll tell everything. So one group, try. I will give you.
Glycerol?
Total will be 200. Because already BTH 100 I am adding. 100 plus, okay, let's take.
100 plus inhibitor.
Okay, I will add. First I want to add thylakoid. Thylakoid is 142 you told. But we will make only 50. So half. 142 half. 71. 71 plus. Next. BTH 100. 100 plus. Next what I told?
Inhibitor 10.
Inhibitor 10. plus
Beta DM 20.
20. Okay, now 201. Okay, 71 is also 2 less. Like 70 microliter, even if you take also, it is like volume is too much because your concentration is very less. But that's okay. In this case, you don't have to add any resuspension or water. This is already 200. But in my case, you see, I have a thylakoid concentration, like I just need to take 26 microliter, which will have 100 microgram of chlorophyll. So in that case, 26 plus 100 BTH plus
10.
20 beta DM plus 10 plus 156. So what I will do? I want to make everything same. Minus 156. 44 I should add either a buffer or water. I should add. This is what normally I do. But as we didn't get that much concentration, we are doing today very less. Understand? And do. Now what all need to be added, you got very clear idea. Now you people can add.
It's totally
Hmm.
molar.
This is my conditions. You should not write this.
I mean, this total.
200. Hmm.
I mean, so it's the same 71 plus.
Yes, yes. 201, that is fine. But you should not compromise like this when you do your actual experiments. Okay. 71, how we got? Not here, not here. Don't look at this.
What did we add? We get 71.
71 because your concentration is like 142 microliter you need for 100 microgram of chlorophyll in whatever you isolated thylakoid. But as it is too high volume, we are making only half, 50 microgram. So total 200 microliter preparation, how much chlorophyll is there now? If I am taking 71 volume.
0.5 micro. 50.
No.
50.
50. And I want to load in one well 10 microgram. How much I should take from this 200? In 200, 50 is there. So I need to take 10. Then how? 2.5. 200 microliter contains 50 microgram. If I am 5 microgram, I can take 20 microliter. If I have to 10, it will be 40 microliter. You can't load 40 microliter in a well. The maximum capacity is 20 or 25 is maximum. So today what we are going to load is also very less. You may not get like that dark band. You will get a very light band because you are loading very less photosystem. Understand? So these are the kind of problems will happen when you do neti pitch. So we will load today maybe 5 microgram, so that 20 microliter you can load 5 microgram, but mostly people do 8 microgram 10.
So 20 microliter out of the thing.
Yes, from the 200 you can take. Okay. So why nobody is ready to prepare? Just prepare. At least 1 or 2 vials. You have enough pipettes also. You can take the things from here and prepare. You also take one. Maybe you also take one. 200 microliter is one more here. You got 20, you got 200. That is 10. Yes, do. So first add your thylakoid. One thylakoid is here, one is here. Maybe one more I will bring. You can add.
We can't.
Nothing. No. Over. Over. Done. Gaya. Japan. Mammies.
No, my God. No, my God.
Who didn't get the sample? This is the sample we stored that day. Take. One more you need, no? You keep that like. And one more thing, I am taking this from -80. You need to keep at least half an hour inside ice so that slowly the ice crystals will like what it is called.
Thawing.
I forgot that. Thawing should be very slow process. You should not take out tap like normal protein we work. You should not do that. Very slowly. Again, gentle. What happened? But the sir was asking to.
Why is Sunday?
He told today is the last day.
Today was the
He's giving a record.
Detector on and see. You know that part, right? All clear for preparation? No need of any help, right? Huh? It's still, yes. It is ready. Ready. I kept half an hour before. And again, when you work, no, you should be like this. Everything in ice, open in ice, tap, again ice. When you are taking also, take like this, keep, you should not expose. This is how you do.
Only one vial, na? Some body. I mean, for
This is sufficient for all of you, these things.
I should
I
load
gave,
only one.
yes, only one is in it. Now 3 will be there. Loading, I think you have 20 wells. You all can load. Okay. You will see the stacking, removal, and also you can do the clipping.
What?
I mean the tank things.
20. It was smooth.
Here, prepare and keep there. Once they prepare the sample, we will bring here. Now, for now, just make things and keep it there. Main thing is removal of the comb, or maybe there are people who didn't see SDS page also. Maybe we will show that also.
You want me to bring that there?
Bring that and show them also. Removing, keeping. 71 thylakoid all added, right? And beta DM you should add last. Once you add beta DM, your solubilization time is starting. So you should count. Add only
I think it's less than 71.
less than 71 it is coming. Oh, take.
We got our total.
Okay. This is how much it is? 71. Okay, fine. Just for demo, you use that. Maybe from this cap and all you can take. Okay. Because again, if I give, it will take a time to thaw and then use.
Whatever is on the wall.
Yeah, take whatever you have. Once they add, we will show here.
I'm keeping it here.
Or you all can turn one time around like people who didn't see SDS page also, they can just, she will show how to keep the tank preparation and all. Yes.
So first we add the dissolving gel, which as she explained, it's a gradient, 3.5 to 5.5. Then we make the stacking gel. After 2-3 hours, we allow it.
Give me a little bit.
And then you can see that the wells have been created there. You can try now.
Hmm.
You want to try?
One more. Just gentle motion. If anyone wants to try with the second,
Who never did SDS page or something, try once.
If you do.
Yeah. So everybody can see the wells right now. The well, yeah. There is separation and small wells are there. The capacity will be like in this maybe 20 to 24 microliter. More than that you can't add. Now you need to fix this both gels on a tank.
Happened, already happened. He was in the room and in the
Pouring later, this one you use her old one so that they can see at least the well.
one floating inside. മൂന്ന് കൈ
This is the worst thing that happened.
ഇത് കല്ല് കല്ല് വെച്ചു
Hospital they went.
എയർപോർട്ടിൽ പോയി അത്ര കണ്ട അത്ര പ്രശ്നമൊന്നുമില്ല
Why yesterday she showed me. Cathode is there. Add 20 mg. To take 200, add 20 mg. ഇല്ല
അത് കാര്യം പറയാം
ഇതെങ്ങനെ ഓണാക്കാം?
മേലെ ഇതാ ഇങ്ങനെ മുകളിൽ
മുകളിലാണോ താഴെ?
ഇവിടെയല്ല
ആ.
50 BTH 40 is there, right? 5
Yes,
ml.
yes, it is there.
5 ml, right?
Yes, yes. You add them give water. But B30 don't add. B30
How to mute it?
Once you add
One touch will you mute?
time is
No mute. No, it's off. It will off. It will off.
Which is off? This one.
ഓപ്ഷൻ കാണാല്ല ഇത് മൈക്ക് റെക്കോർഡിങ്ങിനുള്ളതാണ്
അത് തന്നെ ഓഫ് ആക്കിയാൽ മതി. അതാണ് ഓഫ് ആക്കേണ്ടത് മെയിൻ ആയിട്ട്.
അതല്ല
ഇതാ ഇപ്പൊ ഓണാണ്. അതിനോ അത്ര എനിക്കറിയടാ.
small down
യെസ്. ആ
till here. We'll add the dissolving gel
യെസ്.
and put the comb and add stacking gel. Totally we put the comb and first we add.
No. You first add, then you put the comb.
That's what.
ചേച്ചി ഇവരൊക്കെ ഒന്ന് പോയിട്ട് സ്വസ്ഥായിട്ട് പറഞ്ഞു തന്നെ
അതാ. യെസ്. This is on.
ഇതാണ് ഞാൻ കേട്ടോക്കണം
ഞാൻ ഒന്ന് ക്യാമറന്റെ മുന്നിൽ പോയി നിൽക്കട്ടെ.
ഇത് ക്യാമറ നോക്കണം ഇത് ക്യാമറ
Side pose.
കുറച്ചുകൂടെ അവിടെ നാടകം
ഭയങ്കരം തന്നെ. എന്നാലും അടിച്ച് അയാള് കരഞ്ഞ.
വയസ്സായില്ലേ എന്നിട്ട് ഈ ഒരു കല്ല് ഒരു കല്ല് മാത്രം കല്ലിച്ചു അപ്പൊ ഞാനാണേലും വണ്ടി ക്യാബ് ഓർഡർ ചെയ്തിട്ട്
അങ്ങനെ പോയി.
എയർപോർട്ടുണ്ട്
ഐസെങ്കിലും വെച്ചൂടായിരുന്നു.
ഐസ് വെച്ച് ഒരു ഐസിന്റെ വലിയ പാക്കറ്റ് മുമ്പിൽ കൊണ്ടുപോവാ അവിടെ ഡിസ്കസ് നമുക്ക് വേണ്ടത് ആ പാക്കറ്റ് അതിനുള്ളിൽ കൈ വെച്ചിട്ട് പോകും
We need 100 micrograms of micronutrients. What we will do? It is coming 0.7. Right? How much we will take? That's the matter. Okay, no.
No.
0.7. What is the one?
From the color. This is not the color.
It has to be aesthetic. See, it's colored symbolic.
Yes.
Actually, we keep stirring.
Still, it will come. Like if you put your pet. That only you put, no?
3,250 to
Huh?
580.
I don't know why it is not coming.
And the stand placement, don't ask. I will break that.
Which one?
What is this? सीधा सीधा रखना.
You mean the clipping?
Ah.
I will do. It will come, I think.
It will come big.
Put one bead and keep somewhere. It will come.
വെള്ളാവോ?
അതെ അത് ചുമന്ന കളറായി
I
മാറും.
know that.
ശരിക്കും ഇത്
It
കളറാ.
will become red. Brick red color.
ഞാൻ ട്രോളാക്കും.
അത് വന്നിട്ടില്ല. ആ
This box is like
this box is who gave this box? എന്താ ഇങ്ങനെ ഉണങ്ങിയ ബോക്സ് കൊടുത്തേ? Give them another box. ഞങ്ങടത് എന്നൊക്കെ.
ഞങ്ങളെ പേരൊക്കെ ഇതിന് എല്ലാവർക്കും അയച്ചു. അപ്പു സാറെ വിളിച്ചായിരുന്നല്ലോ.
But video is not there.
ഇല്ലല്ലേ?
Only audio is there.
നടന്നു നമുക്ക് ഉണ്ടാക്കി എടുത്തിട്ട് ഒരു കോമഡി ഷോട്ട്.
It's not running.
method file I used to use.
Not method. No method file, nothing. Just it's running. No.
No.
Why this is not moving? It's going.
This one, na?
Mm.
Fine.
It's so straight.
It's fine.
Let it go. Fine.
चलेगा.
You keep acetone.
आज में
Oh, acetone डाला हुआ है.
Go and sleep. Put acetone. Go and sleep. Your eyes are like this. What you people are adding now? What all you added? Thylakoid? BTH added? Then? I told beta-DM not to add. Okay? Beta-DM, once you start, your solubilization time is starting, which is very important. You can't do like that. Namrata, this is sample.
Small.
Oh no.
Just one.
One is enough.
Two eyelid.
No, to take. I don't need that. Take whole sample with you. Namrata, you will use this. It's getting contamination. Or can you remove that? Remove because it's a contaminating coming. And keep soap water or something. Oh, 1 minute, can you people just, whoever didn't do this SDS, I will just show this, no? How to remove, how I am doing. If you want to see, otherwise I'll do. So, tomorrow just one more experiment, done. People are free and I am also free. You want to see that? Okay. So, this is today.
If time is there.
Let's see. We'll show tomorrow. Or if time is there, let's see. So, this is, you are going to pour the cathode buffer. Okay. So, in cathode, and you can see how the electrodes are connected. Here, for the current pass.
Doing normal SDS.
No, we are doing native page. But you can use the same setup. But your buffer, gel, everything is different. But, that's it. Now take the other, open.
Sides.
Yes, yes.
For this, no dark condition is required.
I told already everything should be dark. What is the importance of dark? No. Srija should tell. It is for photosystem complex. For gel, why I will?
Super complex.
Then should I do this gel also in dark? Should I? Then for what? Huh? For what? ഞാൻ തോറ്റു.
Okay.
Okay. Sheet. One. Just one. And cathode also. Cathode. Our sample is in ice only, right? Sample plus whatever.
You make the buffer maybe?
Yeah, blue one. So cathode buffer I will add. We will just check whether it is leaking. If leaking is there, then the running will not be proper. Current passage will be different, right? If something is leaking, this only taking the current. If that is leaking, obviously the gel running will be affected. It will be like, it should go straight, it will go like this, and it won't be proper. So let's check whether leakage is there or not. If leakage is there, you should again open and clip it properly, and this part of gel is now exposed. It will get dry. If no buffer, nothing is there, this will get dry, right? So for that we will pour and we can keep in fridge so that the gel will be proper. So I think there is no leakage, nothing. So we just, okay, Shreya, just take this, pour and keep it inside. That's it. And anode will go here, and we will put it inside and connect to the voltage. That's it. Now let's prepare. And
Ma'am, the inhibitor is 10 micrometers.
I will give you. So you people, 15 people. How many?
Just 3 people.
No, loading you all can do. Who, like there are people who never did loading and all, no? You people can just load each by, like, well, just load, okay? Prepare. Everything is added? Here also? Yeah. You also added, right? Oh, still it is. You make like this. We are just doing demo. Do.
Beta DM needs to be added.
Beta DM, same time we will add. Then we will see. And during the solubilization time, 3-4 times you can just mix it properly. So that the micellar formation and all, you can just mix it homogeneous.
One do.
No, 3 people do it together so that we can take together to the.
Should I the proportion?
No, now. After adding beta DM, you can mix. Now you can just tap it. It is mixed. Okay, just keep like that. Once you add beta DM, start mixing. And keep the timer also 15 minutes. But 3 of you people start together so that we can go together to the centrifuge. Then loading, done.
Yes, ma'am.
What? Why are you laughing? I'm seriously finishing the experiment.
Done.
I am seriously finishing the experiment today. Yeah.
You're teaching very properly.
That's what I think they are doing it very properly.
I will go and I will listen to this
You
again.
could start
Yes.
a blog, I think.
They will.
The inhibitors are like
Give inhibitors.
I gave already, right?
One more you give.
How much you are adding?
10. 20 I gave, 20 microliter. So 2 people can use. One more I will give. Only 10 is there. 20 is not there.
20 should be,
Sure,
20 should be there.
20 should be there. The stock manager telling 20 should be there. It is not there.
Okay, I'll bring another one.
Huh. See, it is there, but you need to spin. Spin one there. This one. Open it. Keep it. Connect it.
दर्द जोर है
Keep it. Keep this. Keep somewhere. Oh, this is big for that. Give me. Another is there.
this is okay 500 megabits is more
No.
will I
Correct.
run for 1 year
Yes. This is next year sample also. Keep it minus 20. Spin it. Immediately. Spin it immediately so that they can use.
वही कर दो नहीं तो no I
See whether I loaded 20 of them.
Huh?
5 angler is more than enough.
No. I told them to add 10. Let it be 10. This is
What?
nothing.
Understood.
എങ്ങനത്തെ
It's under
അല്ല പിന്നെ
പാട്ട് പാടില്ലേ
പാട്ട് ഞാനൊരു പാട്ട് പാടാം. പാട്ട്
ଦୁଇ ସାରା ଇ 20 ରୁ ଏଟା ହଉଛି 2 ରୁ 20 ମାଗିବ
you are teasing him.
no
Then
yes she told me that then something like ଏଟା ହଉଛି ନାଇଁ ନାଇଁ ଏଟା ମୁଁ ଏଟା ବି ହବନି ଏଟା 200 ଏଟା କାମ କର ଏଟା
done.
this is 10 microliter only na this is 20
2 to 20 maximum. So this is 10. Yes. Ready? Let's add beta DM. And someone take 15 minutes timer. Keep in your phone.
font again I spun this
Is there
ready
any-
for you
Yes. Shreya did a good job. Suhail, can you bring that one for BN page?
Sure.
Inhibitor.
it is already
Extra.
ഇതാണോ ഇത്
close it now it's done
Close.
just red
Red game.
red this side is
for recording
So right now it is on or off?
not recording this
you need to come on Sunday why you will come
That is also true.
She's no timing now.
Do Monday then, Monday morning.
She herself
But-
told that she is going to some her relative on Sunday, Saturday, Sunday, Monday. She won't. Okay.
Then you
All of a
alone.
sudden she didn't. We'll do it on Saturday. I don't know. Yeah, I made her clear that.
Why sir is not directly telling you the PAM work? Hmm. Recording.
You can switch it off.
But how?
Take it out. It's recording. Huh?
It's recording now?
How to stop recording? No. This is the ask him.
Okay, let's start. Who is keeping timer for 15 minutes?
ഞാൻ ചോദിച്ചിട്ട്
Shogi.
ആ അവിടെ ഹൗ ടു മ്യൂട്ട് ഇറ്റ് ടു ടൈംസ്
I think you'll have to turn it up. It's there. You can do it from there, but not from here.
ഓക്കെ
You can turn it up.
എക്സ്റ്റെൻഡ് ഔട്ട് great then
Yeah, now it's
now it's working ഇതില് മ്യൂട്ട് ഓപ്ഷൻ ഇല്ല
Good.
നിങ്ങൾക്ക് ഓഫാക്കാൻ നെക്കി പിടിച്ചിട്ട്
No, it is done.
yeah.
After putting betadine for solubilization, 15 minutes centrifugation, this step should be repeated twice or not.
No repetition. Directly. Guys, in 15 minutes, 5 minutes gap, 3 times you mix it. Inside ice. Okay? With the pipette.
It is a set
Yes.
4 degrees, 1 hour more.
Maybe. 15 minutes, 10 minutes. Then load. Done. Then- Oh. We need to load it.
नमस्ते एमएससी देख के आओगे एक बार एमएससी तुमको देख के खुश हो जाएंगे जाओ यार मेरे को देख के तो मूड ऑफ
Who told? You both are.
जान देखो यार
Yeah.
giving food biryani.
Yeah.
Today dinner done.
Next year, I think she will go to teach MSc people.
Yes.
Next year or next to next year?
Next year, sir started sending
Last year only, second year only.
Because you already studied here.
Yes.
I guess
So no, many.
you know many things. You know many things. Like epitome of academics. Can I have?
It will become more.
It is iced tea. Suhail. Suhail. Your name?
what is the time? 2 minutes time. 2 minutes 39.
15 minutes we have. So one time you already.
What about break?
Yeah. We are on a break. No. Wait 10 minutes. We will keep here. Then 10 minutes break is there. Oh, okay. Papa. Guys, you want to have chai or you want to focus on 15 minutes solubilization? They are very dedicated. They don't want. Yes, guys. After 5 minutes, one more mixing. What?
Lamellar buffer. What is lamellar buffer?
You should see today, Google and tell me tomorrow what is Lamelli buffer.
So I was asking, sir.
No, don't tell. Lamelli buffer is a very basic question and you should know
It's
this.
first time I'm hearing, ma'am. That's what I'm asking.
First time. SDS page you did?
Navodaya I saw.
Lamelli buffer is a denaturing buffer which is used in SDS page. If you have a crude extract of protein, you want to denature them and get their primary structure and load, you need to denature them. So you need to use a buffer, and HCl will be the buffer system in that. You will add glycerol for making it bit heavy so that it will go to inside the well. Then the third component will be any detergent. Commonly used detergent is beta mercaptoethanol or DTT, which is for cutting the disulfide bonds so that the tertiary, secondary structures all will be open. And extra you will heat it. Then whatever is not denatured, in heating everything will be completed. And read more about it. More things will be there. This is what I know. And yeah, TrisHCl, glycerol and beta. Yeah, that's it.
Hey, it's about like
If it is 5 minutes, mix again. Mix again. And in next 10 minutes, mix again. And I will see whose mixture is having too much foam. If you are gentle, very nicely done, there won't be any foam. It will be, third group is not working. Third group. You are not mixing. You are distracting them from learning a major technique. Jyoti is distracting the students.
मैं जब देखा ना तो मैं इस तरह की ठीक है by the time they come we finished. So you give them the option.
White.
new in, old out.
Very old.
What can I do?
It's so bad.
I need eyes.
Why he did that? He didn't see.
Yeah. It's not rough, but
Cleaning is happening. You are running.
What cleaning is happening? Yes, cleaning, and I am just seeing, but it's not straight yet. Let's see. Oh my god. Then we will do it tomorrow. And Jyoti, they want to do CD also. If time is there, we can go and do CD.
Tomorrow we have HPLC only 1 hour.
HPLC, once it is running, we can go down for CD.
Sanket you have, right?
Yes.
What with therapy? Who wants to have therapy?
Yes. Guys, 15 minutes.
Want to make faster?
No,
10 minutes is also fine, right?
no. 15 minutes is good for clammy.
You want to organize them?
Yes. Which college?
And Nandana, right?
Okay.
We haven't
Kannur.
interacted till now.
But I know her. കണ്ണൂർ ആരുടെ കൂടെയാ ചെയ്യുന്നത്? ചെയ്തിട്ടില്ല. MSc കഴിഞ്ഞിട്ടേ ഉള്ളൂ. ഓക്കെ ഓക്കെ, കഴിഞ്ഞു.
It's not for free. Meanwhile, if we become and we will use the meal.
Okay.
He was mocking.
She's not able to. She's standing there.
Who?
She's looking this.
Who?
In this.
I guess I will teach her.
What you should have taught in her, I think. It's on. Not on. It's blue color.
So, it's not on. He told some long press.
I'll directly.
I don't know. This is recording now.
I told them.
No, it will come green.
It's
Wait,
not
wait
green.
for some time. Then hold it, press it again, then it comes green.
That's para and anything.
ഇനി വന്നോളൂ, നിങ്ങൾ നക്കി പൊട്ടിച്ചല്ലേ?
ഞാൻ നെക്കി പൊട്ടിച്ചാൽ അത് വരുന്നില്ല, അവനോട് പറ.
Okay, 15 minutes over.
പഴുത്തുരച്ചി പഴുത്തുരച്ചി
Ah. Where is Srija? Two minutes only now for this centrifuge. Now 3 samples you can keep in common ice box and 1 person can bring. We will keep. Why we are keeping here inside centrifuge? For what? Ah. Cell is not there, only thylakoid membrane is there. Then why? What you are removing? Pigment you will remove, then pigment protein complex? Membrane, you are removing the membrane. You will take only the super complex. Huh. You told pulling down cells.
इतना बार करेंगे तो मुंह से नहीं आएगा।
I don't know this much Hindi, you know that? But I understand mistake हो गया. Done? Bring 3. No, 15 minutes over means your time is over. Bring it. Keep in 1 ice box and bring.
5 minutes, 2 times mixing.
Yes, first, then 5 minutes, then 10 minutes, 3 times. കോഴിക്കോട് എവിടെയാ? Marker is there, marker.
Which mark?
Ragendu has one, right? കാണുന്നില്ല, ഞാൻ ഇന്നലെ എടുത്തിട്ട് തന്നിട്ടുണ്ടാവില്ല. That's okay. Show me your preparation. Let me see. Yes. Good. What is yours? See, I will show you. This is foaming. If you do harsh pipetting, it will come like this. If you didn't do anything also, it will, no foam will be there. Bring that. Give me, keep the 2 in this.
Just a second.
No problem, same sample only.
हमारे पे लिख के दे।
You give me in all 3 or?
No.
Okay, where is the other one? I am not giving any gift. Only quiz will give you gift.
तेरा मुझे लिखा है।
Okay?
7 is 1.
Let's keep. Come. Keep. Open. Remove the lid. Come here. Properly do. Remove it. Now 3 is there. So balance. 1, 2, 3. Triangle you can keep. 1. Yes. Slowly. Yep. We will switch off all the light when we are doing. We will be doing like in dark. Yes. Keep the other. These all step will be like fast. You see? Is it balanced? 5, 5. Yes. Close it. Close. Yes.
There is close button.
Okay. Now speed we will keep 14. a little bit less, and this will be like 10 minutes and 4 degree. Let's go.